So now we're in the early 1900s, and we've had these scientists that have done a lot of work on figuring out nucleic acids and chromosomes. And we had Mendel and his work with inheritance, and then those others that discovered, all of this genetic information was on chromosomes. And then finally, as we get into the 1900s, 1920s or so, we had scientists that started working on the idea, well, maybe DNA is the genetic material. But we kind of knew the structure of DNA in terms of its nucleotides. We didn't know its exact structure, but we knew it contained these 4 nucleotides. And we knew that DNA was a relatively simple molecule, and at the time, the idea was that DNA was really too simple to encode for our genetic material. So people believe that it was the proteins that were made that actually contained the genetic information. There were 20 different amino acids that we had discovered, and so we knew that there was a wealth of information that was contained within proteins. And so the idea was that proteins were the genetic material. So along comes Frederick Griffith, who was a bacteriologist, and he was the first to show that genetic material could be transferred horizontally, meaning members of the same generation and not just vertically, like parent to offspring. So this is a process that he demonstrated called bacterial transformation. So what happens is that bacteria takes up DNA and it makes that cell different. So he was working with Streptococcus pneumoniae, which we will work with later in the semester. And this particular bacterium causes pneumonia. Now there's 2 different strains, a rough strain and a smooth strain. The rough strain is not pathogenic. It doesn't have a capsule. So when these colonies are grown on plates, they kind of have a rough appearance. And so that's the R strain or the rough strain. And then the smooth strain, they have capsules and they are pathogenic. Remember, last month we talked about how capsules could make certain bacteria become pathogenics, and that's the case with Streptococcus pneumoniae, that the strains that have the capsule are pathogenic, and so these would be our smooth strains. And so what he did is he manipulated the effects of live R strains, live S strains, and heat-killed S strains of Streptococcus pneumoniae. So here is his experiment, okay? So he had the control, which was the rough strain. It was non-virulent. He took it, he injected it into mice. And then his first experimental group, he had heat-killed smooth strain. And then in experiment 2, he had rough strain along with the heat-killed smooth strain. And then in experiment 3, he had, again, the smooth strain, but had those that had been recovered from experiment 1. And so here's what happened. With the rough strain, the mouse lives. With the heat-killed S strain, the mouse lives. Okay, the bacteria has been killed with heat, and the mouse lives. But if he takes the rough strain plus the heat-killed S strain, the mouse dies. And then he takes the S strain from the second experiment over here, and the mouse dies. So the combination of the heat-killed S strain and the live R strain causes the mouse to die. And then the strain recovered from this dead mouse here then causes this mouse to die. So he's like, okay, something's going on here. Why, when I put the 2 together, the mouse dies. If they're separate, mouse lives. But when they're together, the mouse dies. So he couldn't figure out, what was going on. And so he was like, okay, something passed from the heat-killed S strain into the R strain. And that's where he came up with the idea of transformation. It transformed the R strain into an S strain, and it was able to kill the mouse. So, a few years later, Avery, McLeod, and McCarty came along, and they wanted to explore this a little bit further. So they isolated that S strain from the infected dead mice. They heat killed it, inactivated various components of it. And so what they came up with really was an elimination study. They used different enzymes that degraded the various components in order to determine exactly what was causing this transformation. So here's what they did. They used heat to kill the S strain of the S pneumoniae and remove the capsule components from the solution. And so we have this color coding here. We have the proteins in green, we have the DNA in blue, we have the RNA in red. Okay. And so here's our control. No enzyme is used. All components are present. DNA, RNA, proteins, all present. In this first sample here, they used proteases, and proteases break down proteins. So the RNA and DNA was still present. In this experiment, they put in ribonucleases. So it degraded the RNA, the ribonucleic acid, but the proteins and DNA were still present. And then the last tube over here, they put in deoxyribonucleases. And so that degraded the DNA. We still had proteins and RNA present. They then took these and added the R cells to each one of these. And so in the first one where we had all three, transformation occurred. Okay, they were able to isolate the S cells. In the second one, transformation occurred. So this showed that transformation could occur in the absence of proteins. So remember, they thought that proteins carried the genetic information. And this test showed that we got these smooth strain cells even in the absence of proteins. So then in the next one where we had proteins and DNA, transformation occurred. So it occurs in the absence of RNA. But in the very last one, where we had no DNA, we didn't get any S cells. We didn't have any of those pathogenic cells, so transformation didn't happen. And so their conclusion was transformation does not happen without DNA. They said DNA then has to be the genetic material. People didn't really accept it because they said, again, they thought that DNA was too simple to be the hereditary material. So Griffith demonstrated, that something was transforming these, non-pathogenic bacteria into pathogenic bacteria. And then Avery, McLeod, and McCarty followed up on that and figured out that it was the DNA that had to be this transforming material. Now, a few years after Avery, McLeod, and McCarty published their results, we had another team of scientists, Hershey and Chase, and they performed their own experiments with this, and they confirmed the idea that it was DNA that was the genetic material. So they were studying a particular bacteriophage, which we know is a virus that infects bacteria. And they were studying the T2 bacteriophage, which is a virus that infects E. coli cells. And this phage will attach to the E. coli cell and then inject its genetic material, its nucleic acids, inside the cell. And then those replicate, and the cell bursts and releases these bacteriophages. So what they did is they took the protein coat in one batch and they labeled it, okay, or they basically injected this radioactive sulfur into it. Sulfur is found in amino acids, but not in nucleic acids, so not in DNA. And so and then they took another batch and they used radioactive phosphorus to label the DNA, because phosphorus is found in DNA and not in proteins. And so they had this batch with the sulfur that was basically radioactively labeling the proteins. And then they had another batch that had the radioactive sulfur, which was the DNA. And so they got some E. coli cells and they put them in separate environments, and they allowed each batch of the phage to infect the cells separately. Okay. And so then, after infection had occurred, they took each batch into a blender where they could basically spin it down into a centrifuge. And what they did is this. So once they basically spun everything down, okay, so here's the bacteria, or here's the viruses, they're infecting the bacteria. So we've kind of got this glowing blue phosphorus DNA here and our glowing protein coat here. Okay. The cultures were spun down to separate the virus from the bacteria. The bacteria is heavier, so it settles to the bottom. And what they found when they looked at the bacteria is that the bacteria infected with the phage containing the DNA, or the radioactively labeled DNA, produced radioactive phages. But the phages that had the sulfur on the protein did not produce any labeling. There was no radioactivity in the phages. So what they found was that only the DNA is what was being injected, and so therefore the DNA is what has to be the genetic material. And so this was published in the 50s, and this really cemented the idea that DNA was the genetic material.